Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Welcome to our class. Today we will be discussing about the role of DNA in protein synthesis. But before we discuss the subtopic of today, I want us to take this moment to thank the Almighty Allah subhanahu wa ta'ala for the incredible power He has bestowed upon us, as well as for the powerful source of knowledge and wisdom He has given to us as the creatures of this world. Besides that, I want us to ask the Allah subhanahu wa ta'ala to make our things easy. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. Now let's class continue and start diving into our subtopic of today. And today, as you can see, I've changed the format of delivering the lesson after getting some concerns from the learners, because the people who are receiving the lessons matters a lot, and therefore they requested whatever thing I'm discussing, I just put on the board first, then we discuss in details. Now, that aside class, today, as you can see on the board, we want to navigate through that subtopic. And as the first thing I want us to discuss is about the significance of the DNA. Now class, what do we know about the significance of the DNA? The DNA is significant in that the DNA is responsible for the process of protein synthesis. Now class, how is the DNA responsible for the process of protein synthesis? We are going to discuss that on later. But the significance of the DNA is that it is responsible for the process of the protein synthesis. And again, class, what is the importance of these proteins in our cells? What we know, class, is that the proteins in our cells, they control nearly all aspects of the life of a cell. And therefore, the proteins are regarded as very important since they control every aspect of a life of a cell. Therefore, the DNA, being a molecule that is responsible for the protein synthesis, therefore, again, takes part in the control of not directly but indirectly, the control of every aspect of the cell, because whatever the DNA is synthesizing is what is enabling controlling every aspect of life of a cell. Class, if I go the next thing, the types of proteins. We have two types of proteins. That is, we have a structural protein, we have structural, and we have functional. Class, if I just start with the first one, that's the structural protein. The structural protein class contributes to the structure of the cell as well the entire organism. For instance, in the structure of a cell membrane, we are going to see that it's made up of two protein layers and a phospholipid layer. That is a case of the structural protein. Let's look at an issue about the functional protein class. Functional protein class is a type of protein, e.g., the enzymes and hormones, and basically they control the functioning of the cell. And therefore, class, if you still remember that we said enzymes are organic catalysts which are protein in nature, and they catalyze the rate of metabolic reaction in this case. Now, again, class, I want us to look at the sequence of bases along the DNA strands. Class, the sequence of bases along the DNA strands, class, contributes to how the bases are linked to form a particular amino acid. For instance, class, in the base sequence, we have the adenine, thymine, guanine, and cytosine. Those, we have four bases along the DNA strand. And again, the sequence of the sequence of bases along the DNA strand class contributes how the kind of amino acids to be manufactured. And again, those amino acids, when they are linked up together, they again translate to proteins class. But again, on the issue regarding that, we're going to look at it. How are these proteins linked up finally to make about the proteins the amino acid, and finally the proteins. Class, what we know about this is that the bases along the DNA strands, they link up in form of a base triplet. They join in form of a base triplet. And a base triplet class consists of three bases, that is either. And the bases here we are talking about is about how the ones that are responsible for protein synthesis. Now class, in the process of protein synthesis, we are going to see what happens to the double helix of the DNA strands, the other molecule that will be involved in the process of the protein synthesis class. Now class, as you can see, we have 4 bases which are used for process of protein synthesis, and these 4 bases are, we have number 1, the adenine. Number 2, we have the next one is called thymine. We have guanine. We have cytosine. Just now class, in the process of protein synthesis, what happens is this thymine is going to be replaced. This is the bases along the DNA strand, and therefore the base thymine is going to be replaced by another base called the base uracil. Examples of codons that code for amino acids, class, the number of bases along the DNA strands, we have 4 which are repetitive, and the way they pair we have looked at in the previous class. Now today I want us to look at the number of codons that code for amino acids. Now class, the bases that code for amino acid essentially are 4, that is adenine, guanine, uracil, and cytosine. Class, you can see that the base thymine was replaced by the base uracil in the process of protein synthesis, and therefore the ones that are going to form the proteins, or rather the amino acids that will eventually link up to form the proteins, are these 4, the adenine, guanine, uracil, and cytosine class. Again class, I want us to look how these bases link up and finally form the amino acids. That is what I want us to look at. This class, we say that these bases, there are 4, and these are the ones responsible for making the amino acids, and hence those amino acids joined together are, or rather condensed to form the proteins. Now what I want us to look at, how are these bases linked up to form a single amino acid? Now class, we say these bases, they are 4 in number, they link up in form of triplets. They link up in form of base triplet. That is, 3 bases link up. For instance, we have a base like adenine, linking up, they are going to form an amino acid called the lysine, as you can see here. When 3 adenine bases link up, they form the base, the amino acid called the lysine. When we have 3 thymine link up, they form phenylalanine. This one, the valine as well. Now class, these triplet bases that do not code, you have them bases that do not code for amino acid, or rather they act as signals that prevent or rather stops the process of protein synthesis. These are, number 1, a base triplet like this, the UGA, uracil, guanine, and adenine do not code for an amino acid. Another one, the uracil, adenine, a base like that do not code for amino acid again. This one, they do not code for an amino acid despite being along the bases of the strand. Why? Class, again, we want us to look the total number of codons formed. The total number of codons formed. Class, the total number of bases, here we have 4 bases. Here we have 4 bases, class. We have 4 bases. And again, we say these 4 bases, they link up in form of base triplets, codons. 3 among these are going to form a codon. 3 among these, again, they are going to form a codon. 3 among these are going to form. Now, class, the total number of codons, how are we going to get? Now, we say the bases, the number of bases, we have 4. And again, the number of positions required to form a codon or rather a base triplet. We only require 3 among the 4 bases to form a particular amino acid. And therefore, this is the the number of bases, 4. Now, the number of bases raised with the number of positions required to form a codon or rather a base triplet class will give us the total number of codons. In this case, we have 4 power 3, and 4 power 3 class means 4 times 4 times 4, 3 times. That will mean 4 times 4 times 4, we're going to get 64. And class, To add on that, we have 64 number of codons, each one coding for certain things. And out of the 64 codons we have class, 3 do not code for an amino acid. And those 3 that do not code for an amino acid class are the uracil guanine adenine, that is a codon, the uracil adenine adenine, another codon, the uracil adenine guanine. And you can say, for you to easily remember, you say, you go away, you are away, you are gone. That is a very simple way you can remember these. You go away, you are away, you are gone. Those 3 codons, or rather those 3 base triplets, do not code for an amino acid. Now, out of the 64, 61 code for an amino acid. 61 code for an amino acid, for an amino acid class. Out of the possible 64 codons, only 61 code for an amino acid. The rest 3 act as stop signals to the process of protein synthesis. And therefore, those 3 are the ones I have highlighted them here, class. Now, class, again, another important thing is, where do the process of protein synthesis occurs in the cell? The process of protein synthesis, class, takes place in the ribosomes. And these ribosomes, class, they are found in the cytoplasm. They are also found on the endoplasmic reticulum as granules, hence are called the rough endoplasmic reticulum due to their possession by those granules called the ribosomes, class. Again, class, here we have 3 things which are related. That is the nucleolus, ribosomes, the nucleolus, ribosome, rough endoplasmic reticulum, as well the proteins. How are they linked up, class? Now, class, what we know is that the nucleolus is the site where these ribosomes, which are either found in the cytoplasm or rather attached to the surface of the rough endoplasmic reticulum, are being manufactured. And therefore, we say that the nucleolus is the tool that provides these resources. For instance, these are what the ribosomes. Ribosomes are being manufactured from the nucleolus. Class, again, what is the essence of these, ribosomes? Now, class, these ribosomes, they take or rather receive information from the DNA by the messenger RNA, and they start synthesizing proteins. And therefore, we say the ribosomes are a site for protein synthesis. And again, class, another thing that you also need to understand again is that the proteins being the site of protein synthesis, what about the protein? The protein now is the product formed by the ribosomes, and that is how we are going to link the 3. We said nucleolus manufactures the ribosomes. The rough endoplasmic reticulum, class, provides workspace, or rather the surface, onto which these granules or ribosomes are attached to itself, and we say the proteins are the final products for them, the ribosomes. And that is how protein synthesis the ribosomes, nucleolus, and the rough endoplasmic reticulum are interrelated. Class, again, I want us to look at something very important here, and that is how are these codons, or rather base triplets, are being transferred from the DNA into the cell, and what are the nucleic acids, or rather molecules, responsible for these. Class, what we know is that the DNA acts as the parent template. The DNA found on the double helix acts as the parent template. And again, class, we have discussed about the process of the DNA replication, the unzipping and unwinding of the double helix. Now, what happens, class, is during this one, when we want to, the DNA gives out the instructions regarding to which type of amino acids to be synthesized by the ribosomes, and various amino acids joined together, they're going to form the protein, class. Now, class, for this information, and we know that the DNA is basically confined in the nucleus. Now, there must be a means of communicating the information from the DNA to the ribosomes so that it can give the information regarding to which type of amino acid, hence the protein, to be synthesized by the ribosomes. The ribosome synthesizes proteins, but it requires information on which type of protein to be synthesized to get it from the DNA. And again, we know the DNA is now confined into the nucleus and cannot move out of that. And therefore, there has to be a means of communicating the information from the DNA to the ribosome onto which type of amino acid to be synthesized, and hence the protein class. Now class, what happens here is that the double helix of the DNA and zips, and when the double helix of the DNA and zips, the two strands align themselves, the nucleotides of the two strands align opposite to one another. And this is what we are speaking about class. Let's say the double helix was like this class. Now what happens during, we want to transfer information from the DNA to the ribosomes. What happens is the double helix of the DNA strand class unzips, and the free and the two strands align, and the bases and the nucleotides, or rather the bases, the nucleotides, or rather the bases, align themselves opposite to one another. And therefore, another strand will be formed at the middle, and this strand now will copy from the parent template, that is the parent DNA. Now, in the formation, now class, whatever is going to be formed here is called the ribonucleic acid. Is a molecule of a nucleic acid that again is going to transfer the ribosome, the information from the DNA to the ribosomes. Now class, during the formation, that is why we are calling here the formation of ribonucleic acid. Now class, the ribonucleic acid is basically formed when the double helix of the DNA unzips, and the nucleotides, or rather the bases, align themselves on the opposite sides of the two unzipped strands. And again, another strand will be formed at the middle. Now the strand will copy from the parent template, in that it's going to form the base pairs, just as we used to know. But what happens class is the base diamine is going to be replaced by the base uracil, And class, when the base diamine is being replaced by the base uracil, an RNA is formed, and that is how ribonucleic acid is formed. Ribonucleic acid is basically formed when the double helix of a DNA strand unzips, and when the double helix of a DNA strand unzips class, the nucleotides align themselves on the opposite strands, and another strand will be formed at the middle, and that strand will be copying information from the parent template, but it's going to replace the base diamine with the base uracil. And that strand form is now what we call the messenger RNA strand. And the messenger RNA strand is now just a template of the parent DNA, in that it only replaces the base diamine with the base uracil. Now class, next we have types of RNA. We have two types of RNA. We have the messenger RNA and we have the transfer RNA. Now class, what does the messenger RNA do? The messenger RNA class transfers genetic information from the nucleus to the cell. That is concept is very right. The transfer the messenger RNA transfers information from the nucleus to the cell. Good. And that is how it is. Now, once the information from the nucleus is taken to the ribosomes, what happens next is class, the next thing is this information will be interpreted, or rather translated, by the ribosomes. And therefore, once the information is translated, the ribosome is going to get the type of amino acids that is required to be synthesized by the ribosomes. And therefore, it is going now to send another RNA that is called the transfer RNA. Now the transfer RNA is going to the cytoplasm, where we have a pool of amino acids. Now the type of amino acids that will be taken by the transfer RNA is dependent on the information encoded in the messenger RNA, and therefore is going to transfer amino acids then to the ribosomes. And once amino acids are assembled to the ribosomes, amino acids will be formed, and therefore amino acids are going to be condensed and protein is going to be formed. And class, I think that is very clear about how protein is going to be formed in a nutshell, and that is how things go about. Again, another very important thing I can say is protein molecules determine the inherited characteristics in an organism. Explain. Protein molecules determine the inherited characteristics in an organism. Why, class? Because we know a class is that the genetic information contained in DNA is being translated, or rather interpreted, through the process of protein synthesis. And therefore, because the genetic information are the hereditary materials, or rather the hereditary things that tell us whatever the organism has inherited, and if that information again can be translated through the process of protein synthesis, therefore class, we can say protein molecules determine the inherited characteristics in organism. Why? Because it's very simple. The genetic materials in the DNA are being translated into characteristics of an organism through the protein synthesis. And therefore, protein synthesis, we can see they translate the information contained in the DNA, and the DNA contains the hereditary materials. Class, and that is a brief about the role of DNA in protein synthesis. Now, class, and that is what I wanted us to discuss, and we are going to discuss further things in our next class. But before, I want us just to be brief about the formation of the messenger RNA, class. Now, class, I said during the formation of RNA, what happens is very simple. The double helix of the DNA is going to unzip, and when it unzips, the bases or the nucleotides will align themselves on the parallel strands, on the opposite sides of the parallel strands. Another new strand will be formed that is going to copy information from the parent template, but what it does is it replaces the base thymine with the base uracil, and therefore an RNA is formed. Now again, how is the RNA transferred from the nucleus to the DNA? It is transferred by another type of RNA called the messenger RNA. The messenger RNA transfers genetic information from the nucleus to the cell. Again, once the information reaches the cell, the ribosomes translate this information and now dictates the type of amino acids to be linked to form that particular protein. Now, when the ribosomes translated those proteins, class, what happens that it again now sends another type of RNA, that is the transfer RNA, and the transfer RNA is going to the cytoplasm, where we have a pool of amino acids. Now it's going to take amino acid. Now we said the type of amino acids taken from there, or rather the basis, is dependent on the information encoded in the messenger RNA, and therefore protein synthesis takes place and through the process of amino acid formation, and when amino acids are condensed, protein molecules are formed. And lastly, I said protein molecules determine inherited characteristics in organisms. How? Class, we said the genetic information contained in the DNA are being translated into characteristics of organisms through the process of protein synthesis. And you can see if that process of protein synthesis can translate the information contained in the DNA, therefore we can say protein molecules do determine the inherited characteristics in organism. Wish you all the best. May Allah subhanahu wa ta'ala safeguard us. Ramadan Mubarak.